I'll take this off briefly. All right. Good afternoon, everyone, and welcome to our seminar today as part of our Open Science Initiative here at the Center for Brain Health. Before I introduce our speaker, I just want to sort of remind people about what this initiative is about and some of the things that are going on. So the goal of the initiative, which is what we're calling in a buy-in phase, is supported by the Tannenbaum Family Foundation to explore whether the Center for Brain Health should become what's called an open science institute. And if we do that, we'd be joining other neuroscience institutes in Canada that have already taken this step. So in Calgary, at McGill, and also at Western University, they're going through the same process that we are. So the implications of being an open science institute will affect some ways in the way the center operates, but most importantly, it'll be providing training, resources, encouragement, and promotion of open science activities among our researchers. So because we're in this buy-in phase, part of what we're doing is doing assessments of what people's needs are, like why they might not be already doing open science, what they perceive barriers to be in terms of resources that they might need. So we've had ongoing activities to do that, but one of the things that's live right now is a survey that we put together that we're trying to get as many members of the community, trainees, staff, faculty, to weigh in on how their current approach is in terms of open science and what sort of barriers there are in their opinions about where the Center for Brain Health should position itself. So Jeff is going to put a link, or if he hasn't already, to the survey in the chat for those of you on Zoom. We will be email blasting it around as well to get as many responses as possible. We are going to have other events and activities that are part of the initiative, so watch the space. Before I forget, that immediately after the seminar, in one of the third floor meeting rooms, there's going to be a chance for trainees to meet with Dr. Edwards. So now I'll introduce today's speaker, Professor Alan Edwards. He's a professor of medical biophysics and medical research at the University of Toronto. He's the founder and CEO of the Structural Genomics Consortium. And he's also an entrepreneur who's founded or co-founded many companies, including Borealis Biosciences, Affinium Pharmaceuticals, and more recently, M4K Pharma. And I think he might talk about the latter today. He received his PhD in biochemistry from McGill and did postdoctoral work with Roger Kornberg at Stanford, where in the 80s and early 1990s, he was part of the team and conducted seminal work that was a major reason why Roger Kornberg received the Nobel Prize in Chemistry in 2006 for elucidating the structural basis of gene transcription. So after his postdoc, Dr. Edwards was at McMaster University as a faculty member and later the University of Toronto, where he continued to do pioneering work in structural biology, including the development of the concept of structural genomics, so collecting structures for all the proteins that a organism expresses. So his work and determination of protein structures at large scale makes him and his team, I think, the largest contributor of protein structures to the protein data bank, with over 4,500 submitted. But the reason that Dr. Edwards is here today and why I'm really excited to have him here is that he's a pioneer and leader in open science. So he was a key player in the development of the Open Science Institute that was taken up initially by Guy Rouleau at McGill and is spreading across Canada. And again, we're trying to look into doing that here. I also learned in researching his background that he was a developer of the open lab notebook concept, which some of you may have heard of, where people don't just publish their research when they're done with it, but actually make their lab notebooks available to people as they're doing their work. It's a very interesting and innovative idea. And so most relevant to today's talk, he undertook a really innovative approach for the Structural Genomics Consortium in its partnerships with industry, where they're developing drugs using an open science model that doesn't necessitate restrictive intellectual property like patents. So he's really thought deeply and written extensively about the potential that open science presents to society, and I'm really looking forward to learning from him today. And his seminar is called Open Science from Genes to Affordable Medicines.
Thank you.
Thanks for being with us.
That was long. Now I got to talk faster. I only have a certain number of slides, so it's a lot of fun being here. I was just telling them our daughter went to school here in about 10 years ago, I think, for her graduate work. So the purpose of this talk is not open science. The purpose is to try and have an impact. And as biomedical researchers, let's define the problem that we're aiming for. So around the world, we spend $250 to $300 billion on biomedical research. That's in the public and private sectors combined. And for that, we've cured, we have antibiotics, we have heart medicines, hypertension, statins to lower your cholesterol. But for many diseases, we don't understand yet how they work. So Parkinson defined that disease in the early 1800s. And if I gave you each $2 trillion and I said cure Parkinson's in 5 years, you'd go, I don't know how. We don't really know what the fundamental basis of it is. And until you tell me that, I can't cure it, so I can't promise. And so if you think about a drug discovery point of view, the problem is not, can I make a drug, but to what is the mechanism that I should try to inhibit or not, right? And because when you're trying to make a drug in the absence of knowledge, like the next Alzheimer's drug going forward will be a wild ass guess if it's right or not, and you'll spend $2 billion to find out, oh crap, that was the wrong idea, the mechanism. I made a great drug, but the idea was wrong, because we incentivize that through the private sector, right, where the drug is an asset and not a human right, the business model that we put around it forces us to charge prices that most people on the planet can't afford the new medicines. It's nobody's fault. It's the system we put in place, right? If I'm the CEO of Merck, my job is to make as much money as I can. My job is not to help the developed world, right? And all of us are complicit in it. You probably all somewhere in your stock portfolio or your pension plans own Merck stock. You have every right to go Merck annual general meeting and say, God damn it, make less money. But we never do, right? So tacitly, we society accept this. And I think we shouldn't, right? So how can we get around this? And so first of all, we have to look in the mirror as academics and say a lot of it's our fault. And in this graph here, I'm going to point, right, so the Zoom people can see me. Over here, can you see? Oh, can you see my Zoomy guys? Can you see my? Well, I'm assuming you can see my pointer. Okay, so in this graph here is on this axis is the name of genes of human protein kinases prior to the sequencing of the human genome, where we didn't even know some of these existed. And on this axis is publications. And I rank them from most popular before the human genome was sequenced to the least popular. Now, no matter how you graph this, you're going to get that sort of power law curve. But you can see that most of the activity before we knew about all these genes was on a subset of the kinases. Makes a lot of sense. Fast forward 16, 17 years, and you can, in advance, think, okay, what should have happened? Well, hell, we know we don't know how Parkinson's work. We know we don't know how Alzheimer's work. We know we have a vast knowledge gap in disease. Holy crap, look at all these genes that we know nothing about. Maybe if we understood how all of them work, we'd be able to understand these diseases. Now, that's what makes sense. But that's not what happened. Our incentive systems in academia don't force us to work on the unknown. You cannot write a grant for a protein down here. Right? These are in the same order, right? The same genes that were popular remain popular. And you can just think of it. You train students, they work, you're told, work on something you know, because that's, it's safer. So you work on the same thing your supervisor worked on, right? Try and write a grant on a protein that says, I don't know what it does. I want to find out what it does, period. That grant's going to go in the garbage. It's not elegant thinking. Where's your hypothesis? Where's your this? And so the whole sort of system conspires to keep us working on the same thing over and over and over again. Sometimes they come out of the noise. That's BRAF. It happens to be important for cancer, and there's a drug to it, so everyone can pile on and work on it. There's some Parkinson's kinases down here, but for the most part, 70% of the world works on the same. Now, even worse. So one hypothesis, right, because we're in Canada, this is a world thing. Maybe the advanced countries are more daring in their research and do it. So and they sort of move on the unknown. So you do the search, SARC, you do this search by just going to PubMed, putting SARC, which is the name of a kinase, how many papers, ABLE, how many papers. But if you go ABLE kinase Canada, you get of the total global research, only the papers published by Canadians. And you can see the pattern of how we map on our research compared to the world. And there's no difference. And think of it. If every Canadian got hit by a bus tomorrow, the planet probably wouldn't care, right? If anybody out there says is working on an area where you're afraid of being scooped, that's another way of saying I am redundant, because someone is doing it, right? There's so much unknown in biology, and yet we focus on the same stuff over and over again. How do you get people to work where that box is, right? Where if you had $1 to spend, that's where I'd spend it, because you're going to get far more value per dollar or value per neuron, right, than working on what everyone else is working on. And it's really hard to work down there because you're going to be a pariah. You're not going to get invited to a meeting, right? There's no meeting on kinase 423. There's lots of meetings on ABLE, where all the ABLE people get together and they meet every year and they go from this, oh, where are we going next year? We're going to have it in Madrid. Oh, good. And they get together and they have these little parties and they review their own grants and they have all the ABLE clubs. It's a great way of just continuing the R&D. And it's just the reality of what happens. And the person who works at the bottom never gets invited to meetings, right? Because who the hell would work on kinase 423? It's not important. I say it facetiously, but everyone up knows there's a kernel of truth in all of that. So this is actually why I set up the Sgc. I was doing genomic stuff. I stopped working on polymerase a few years after I came home when the yeast genome was sequenced. And I said, why am I working on why lysine 22 is doing this when there's 4,000 genes that nobody's ever seen before? You can tell I got attention deficit. It's going to be way more fun going out and finding out all these things to do instead of competing on, that one protein. And so we set up the Sgc and we figure, well, if we're going to do this as a community and stop that parochialism like the Canada does this, we need to have our results shared. So if we can work down there, then we can share it with everybody and not have everybody compete on the same kinase 423. We now have about 300 people around the world in 6 different labs, and we work with industry, as I'll show you. And when we do that, we had to create rules of engagement. And if you had to work with multiple people and put all these patent rules in place, you'd be more lawyers than scientists. So we just said, okay, we'll never patent anything. And you'll see the results of it on the next slide. And we publish about 25 papers a year co-authored with folks from Pfizer and Merck and all those folks. Remember, and what's happened over the years, how's the Sgc structured? Just sort of because it was a business talk I put in these slides. So we're a charity based in the United Kingdom. We were founded by Wellcome. We have a board of directors that anybody who gives more than 5 million euros to the charity gets to sit on the board. And they don't tell you what to do. Like, I'm the CEO of the green bit. They just sort of say, here's the right direction you're going, and we report back. And then we have labs in 6 different places, 2 in Canada, Sweden, Germany, America, and in the UK. And over time, we started just doing structures. Structural Genomics Consortium. So we just solve human structures from a list that the pharma gave us. You get any of these. But then we started to use the structures to do chemistry and make small molecules freely available. And the more projects we did under this open umbrella, the more pharma companies joined. And remember, every time you see a dot, that's a company giving us $5 million, well, 8 million Canadian, 5 million euros, right? And they donate it to the charity. We carry out our science mission, and they know all the data is going to be shared with everybody. But because we're working on that unknown proteins, right? The companies say, look, if we let the system do it, they'll work on the same old. We believe there's value out there in the unknown, but it's too expensive for us to do alone. Let's pool our resources together and just explore those proteins on the long tail. And maybe if one comes up, which over the course of 20 years, many have come up, we can start drug discovery projects right away without having to do royalty agreements with universities or blah, 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 blah. We have a so-called freedom to operate that we can just take the knowledge and run. And even if the knowledge is a pretty advanced chemical, it's fairly easy to create a new intellectual property position after that. So that's what our sort of mandate has been, and it's been pretty successful over the years. So I'm going to tell 3 stories now, not about the science, but about the business and sort of spin-offs that one might imagine happens to an institution that decided to go open. And as you know, when the Montreal Neurological Institute adopted our open science policies, they saw a rapid increase in finance and philanthropy, a rapid increase in industry funding, and in recruiting. They found a lot of and patient foundation money. Every indicator, performance indicator of funding, which is really important to professors, it went like that. Okay. So I'm going to talk about 3 projects. The first is these target enabling packages. So how do you actually get a professor to work on kinase? It's a risk. It's a risk because, oh my gosh, there's no cell line, there's no antibody, I got to make the protein. So much of the tools to do science have to be assembled before you can actually do the experiment. So we thought, why don't we do that on behalf, right? So if a protein comes up just because it's unknown or for any kind of these sort of databases, we really want to, if you're talking about the pharma, they really want to get, okay, I want to know what the target is so I can make a molecule and do my drug candidates. In the middle are all the tools. And that's what we've decided to make for a bunch of the proteins. We call them target enabling packages, a drug target coming from the gene sequence. How do you turn it into something where industry go, thank you, I'm on it. And the purified protein, a good assay, antibodies, reagents, right? Chemical matter, iPSCs, all these kinds of reagents that if you're going to go into the lab and say, what does this protein do in kidney cancer? You're going to need all of that. But if we're creating these reagents, you might say, oh, they're very valuable. Like a chemical probe, what we make, it's $1.5 million, right? Why are you giving it away? Well, I'm going to take a orthogonal thing and just give you evidence about patents, and I'm not going to judge. Heidi Williams did a very cool experiment. She took all the genes in the 90s, because remember, the private sector was patenting genes, and they didn't patent others. And she thought, well, that's very cool. Why don't we look at what happened to the business world on the set of genes that were patented and were not patented? Intuitively, you'd think they patented the ones that are greatest commercial value, and they should be the ones that generated the most economic gain. 30% more economic gain from the genes that had never been patented, right? So there is an experiment in the real world, a decade long. You don't have to make models or have hypotheses or insert your bias on it. Those are very blunt experiments. This large number of genes had been patented. This large number of genes hadn't. Let's track commercial outcomes, far more from the ones that were freedom to operate. Indeed, the case against patents can be summarized briefly, said someone from St. Louis. There's no evidence they serve to increase innovation unless productivity is measured by the number of patents awarded. And on and on and on. I'll leave it up there for a sec because you guys can read it. Okay? Let's look at Canadian tech transfer. So in 2010, when the last report from Autumn, which is the organization that watches it, makes sense, published it. Now they keep it secret. Canada spent $10 billion in the research innovation system. Of that, we generated $57 million in licensing fees from the stuff we patented and got it. So $10 billion and we made 57. That's a pretty small number. But wait, it cost us $55 million to generate that 57. And that's a net of $2 million. On top of that, we spent a whole whack of money on these commercialization accelerators. On top of that, we have a whole bunch of money and time delays in doing all these legal agreements in between universities to transfer material back and forth. If you think, okay, universities are an inherent part of our innovation system. If it's important, they'd make money. They don't. They lose money. Our tech transfer system loses money. That's just numbers. U of T is a great case study. We have a $1.2 billion R&D budget at the U of T, a $2.5 billion operating budget. We get, in 2010, this is $87 million from industry. This is contract research. 25 million research income. A lot of that comes from my organization. And that's how much we made from licensing, right? That's the University of Toronto, the largest university in the country. I don't think that's a win. So summing up, there's no strong evidence that patenting an institution drives economic growth. Protecting and facilitating with all these tech transfer things is having a marginal impact on the bottom line. And this is what I like to think about. We don't think about the counterfactual. What happens if we did not do it? What happens if we did share? There's no reason to believe it can be worse. What are we going to lose? $9 million at the University of Toronto? That's not, right? That's like one CIHR Foundation grant, right? What is to be lost by sharing? If you look at your UBC mission statement or whatever it is, I think motto, it's like, it is yours. It's not, it's mine to keep and then you can't have it. University of Toronto is about equity, fairness, justice. It's not make as much money as you can, right? I think we honestly have lost our way in terms of what our role is in society as a public institution. And there's no evidence that doing it the other way actually is doing any good from the economy point of view. The reason for the young people who were in this boat is in the 90s, as research funding was coming down in the country, we said we needed a new way to attract money into MRC at the time, which turned CHR. So we talked about universities as economic engines of growth. And then the policy wonks came in and started to believe that. And that's been part of our university talking points to the government since the beginning. Everything is couched in economic terms. They don't fund science for the betterment of society. Our government funds it for economic growth, and we're just buying into it. And so we're told to patent, we do it, but there's no evidence that it actually does anything. And so for the TEP thing, the target enabling packages, I'll show you what the outcomes have been. So we play up front there on this left-hand side there with industry and academia and us. We make these reagents. We've had organizations, we just got a, I don't know, $8 million grant from the Gates Foundation to make TEPs for non-hormonal contraceptive targets. In Alzheimer's disease, this was a $37 million grant from the NIA, National Institute of Aging, to make TEPs and pre-competitive compounds to novel targets in long tail targets in Alzheimer's. So funders are really starting to buy into the fact that this is a great idea, that sharing this kind of knowledge early, and we explicitly put in this NIH grant, we will never patent. We will generate all the molecules, the chemistry, the lead compounds, and put them in the public domain, and it was ranked #1. There's no, sometimes professors use arguments, oh, the funders, they want, there's this little benefits to Canada section, we need to put an IP thing there, it won't get funded. It's just not true. It's one of these legends of IP. So one of the TEP things we wanted to was an antibody. And here we're going to dive into the science for a little bit. So C9orf72 is a gene that it's mutated in ALS. It's the most commonly mutated gene in familial ALS. It was identified in 2011, I think. Yeah. And lots of papers on it. And as soon as it was identified, people say, okay, what does this protein do? Right? Because all of a sudden we have a new protein, like you can tell it was unknown, chromosome 9, open reading frame 72, right? And so, first of all, you say, okay, if we're going to find out what the protein does, we got to know where it is in the cell. That gives a little hint, nucleus, it might be a transcription factor. And sure enough, in Neuron, they published it's a nuclear protein. So obviously, start building hypotheses about that. But hold it. In Nature Medicine, now it's an early endosome, late endosomal protein. I think, okay, maybe it traffics from the endosome to the nucleus. And then in embo, hold it again. We have the autophagosome. And then patients and families are giving the money to us to study ALS, us, the royal us, thinking that we're doing the right experiments. And 10 years of ALS study on this protein, and we're none the wiser, really, about what it does. And it gets back to the reagents. And so clearly, this is a result of folks using reagents that don't recognize C9orf. And so the solution we thought of, well, the traditional solution which funders have done for 20 years, is start an antibody effort, make a new antibody. But all these folks had antibodies. There's nothing wrong. There's nothing inherently magical about making antibodies. How do you make sure you got the right antibody? Okay, so we thought, well, maybe the antibodies are out there. There's 1 million antibodies that are marketed by the various companies, not on CNI North, but in toto, right? So why don't we say, instead of being the 200th project that makes yet another antibody, why don't we create a project that takes all the antibodies that exist and finds the best one? And so the strategy, which is enabled obviously by CRISPR, is to make a knockout line that lacks your gene of interest or protein of interest. So the cells are identical, except one's missing one protein in, first approximation. Make extracts and then get all the antibodies that are out there, compare them, mano in the same experiment at the same time. Be careful and quantitative, do it under a standard operating procedure. And then if you get the answer, go for it. So this is the antibody that was used in the most common publication that's cited hundreds and hundreds of times. One of those might be C9orf72. This is a Western blot. You guys have all seen Western blots before. Runs around that size, I think. I can't even remember. I don't know. This is the wild type cell. That's the heterozygote. And these are 2 extracts from 2 knockout lines. And that's the one that people are using to find out what the protein does. Well, out there, unbeknownst to us, because it had never been published in a paper, GeneTex had this wicked ass monoclonal. And you can see the signal is exactly as you want, right? The homozygote, the wild type, has 2 copies. The heterozygote has 1 copy deleted, and the 2 knockouts have it gone. That's the protein. This is not. And now you go back to those, immunofluorescence studies I showed you about. Now you're beginning to see, and all the antibodies used didn't recognize the protein. But now that we found one that did, this is Carl and Peter at the Neuro. Now you can start doing the proper experiment. And in this one, what Carl did was a pretty cool experiment. He took the wild type and knockout cells, and he stained one with RFP and one with YFP, and then he mixed them on the same cover slip. Because if you do immunofluorescence, you can play a lot with the gain and with this to see what you want. So you want to normalize or standardize, all the background. And then you can stain with your antibody. Then you look in 3 channels. One channel shows you where the wild type cell is. The other on the same grid, one shows where the knockout cell is, and then the other one. And so in this one, this is a specific signal. You can see it much better on the screen. But what you can see down here is that only the wild type cells label and the knockout cells don't. And so now it turns out it's perilysosomal. The whole purpose of doing this rigorous process is now we can start doing science on this protein. So how to scale it was the business problem. And this gets to the open science, right? You could think, okay, as the American let's create an organization that standardizes and tell industry what to do. Industry says, I don't want academics telling us what to do. So that they tried it didn't work. So you need to bring them on board. But if you bring them on board, how do you bring competitor companies, right? These are all sold by companies. And so it's been around since I was a graduate student, since Babco started to sell antibodies in the late 80s, right? You just didn't know the antibody was good. How do you judge one over the other? It's too expensive it should be professors and you guys who do experiments actually do this. But where are you going to get the knockout cell? Who are you going to have the time to do all this kind of it's really expensive, and you got to publish, right? So someone should do it on your behalf, and doing it all at once makes sense like we did, but who's going to pay for it, right? It's not whose business incentive it is to do that. So what we did was we brought together the vice presidents of these companies to a retreat. It happened to be in Barbados. That's one way to get them there. And we proposed to create an open science effort where on behalf of the community, we would test their antibodies. We wouldn't rank them, just do the experiment. And the companies that have now joined, of which there are 10, you'd think, oh, why would a company join if you're going to show their antibody sucks, right? But these companies really believe in their products, and they're having a tough time marketing it. Hey, I got a good one, like GeneTex. No one had ever bought their antibody. It's the best one out there. because everyone looks in the literature, oh, in Cell, they use this antibody, I'm going to use this antibody, even though it didn't recognize the protein. So it's a great way for them to get their data known. And so we put it in the public domain. So then you think, okay, great. So we got an idea. The companies want to do this. We want to do this. What's the correct business structure for it? You create a for-profit company. Well, the companies didn't like that because they said, what if it's bought by somebody and they start not sharing and then they start to use our data for nefarious reasons? We don't like that. Do it in a university? They say, no way. Way too bureaucratic, and it's not the university's job to do business stuff, and it would take too long. Create a nonprofit? Maybe, but a nonprofit has a whole bunch of rules in getting grant funding. So this is what I did. My inspiration was Paul Newman. So for the young guys out there, he was like Brad Pitt in the 70s, right? And he's wanted a way to give away money. And so he said, he wrote a book, Shameless Promotion of Celebrity for the Public Good. He realized if I put my face on anything, people will buy it. I'm going to make salad dressing. He liked salad dressing, but he didn't wake up in the morning saying, I want to be a salad dressing magnate. It's a vehicle to give away money. And so Paul Newman's own, you've seen it in the shops. It's a company. It's for profit. It pays its employees. It pays its taxes. It does all the things to contribute to the economy, except after the tax profits, don't go to Newman's family. Don't go to shareholders like a public company would. Get gifted up to the charity, Newman Foundation, and it gives it away. And I thought, well, why can't we create a company? That sounds like kind of what universities would like, right? Because if professors work to a cause and there's profit, if somebody gets some of it, they want it. Professors are like kindergarten children, right? Johnny gets more candy than I'm upset. If everyone gets the same amount of candy, we're happy. And so if everyone gets a bagel, we're happy because nobody got more than bagel. And so why don't you create a company where it can work in the common good? If there's money, we give it away. And it goes away to somebody, not back to the university, not really away. So that there is clear, no one is making money off we're donating it to certain causes. So that's what we did. We created the equivalent of the Paul Newman's own foundation. We called it the Agora Open Science Trust. And it is the 100% equity holder in this company. Why? For antibody characterization by open science. And this company does, on behalf of the community, antibody characterizations for targets. And we started in neuroscience. And we're going one at a time. And so the Agora has to be trusted, right? Because if it's going to be given away to somebody, you got to trust that organization. So it was Canadian, so we needed 2 Canadians. So as much as I can be trusted, I'm on it. Mark Krimble, who's the president of the Krimble Foundation, he's a philanthropy in Toronto. Michael Morgan was the UK equivalent of Francis Collins in the Genome Project, and it was the founding CEO of the Sanger Institute. And Else used to be the head of access to medicines for Doctors Without Borders. She lives in Geneva, Michael lives in London, UK, and then the 2 are Canadian. So the idea is, okay, these guys are going to make sure that the money goes somewhere good. Doesn't really what it is, but it's going to go somewhere good. So the science structure, we decided, now this we couldn't have predicted, but this is what happened. We have themes underneath Icarus because it's easy to cluster these proteins in themes. And the neat thing of the business model is that if you want to make a donation and get a charitable thing, you can put the money into Agora and say, use it to do Parkinson's genes. Some grants in Canada are only available to for-profit companies. Icarus is a for-profit company, and so you can get granting schemes, or you can get some foundations like the Michael J. Fox Foundation. These are not theory; these are all real, right? They can give a donation, not a donation, or contribute to the company on a contract. You can get grant funding. So the Sgc got the funding from the Gates Foundation to do non-hormonal contraceptive proteins. And for 8 of them, we need antibodies. So the money can flow into Icarus, or in this instance, directly to McGill, to the neuro, to do that experiment. And then there's Canadian and NIH grants that fund specific projects in the neurosciences. Okay, and so the lab just runs this standard operating sort of procedure. It's agreed to by all those companies. Every month they sit on the board. We go over the data. All these companies donate. They put cash into the company to help fund the organization, as well as they donate all the antibodies that they've made for a given target. Those are some of the funders on the left. It's a pretty cool, it's got about $5 million, I would say, in funding since we started it 18 months ago, just to do this simple experiment. But that's such an important experiment, as you can see from that ALS study, right? You do, and we're focusing mostly on renewable antibodies, because if you do the experiment once, that antibody, especially if it's recombinant, and will be good in perpetuity. You don't need to keep doing the experiment over and over and over again. Here's an example of one. This is a Dynamin antibody that's been used by most of the community. It was developed ages ago by Sandy, I can't remember her last name. And that's the blot, but that's the blot of the purified protein. If you look at the blot of the wild type of knockout cells, that's the antibody that folks have been using to study Dynamin over the years. It's sort of the standard in that little field. And you can see when we tested a lot of the antibodies, there's a lot of good ones out there. So this is a Western blot of wild type extract knockout. And so you can see that some of these antibodies are tremendous and renewable. So clearly, if you work on this protein, this is the data we put on the website. It's in Zenodo. We don't rank them. We don't say, you guys can make up your own mind, but it's pretty obvious, right, which ones. And some of them are mouse and some of them are rabbit. If you need to do different antibody stainings, there's the flexibility, right? The bottom is a Ponzo S staining. That just shows you the protein. That's a loading control to make sure you loaded the same amount of protein in each gel. And there's some, this is a fantastic one over here. There's a knockout cell right here, which you can't see. And there it is. So again, we're going through now, I think we've done about 30 or 40 different proteins, all related to neuroscience at the start. This is Parkin. Parkin is obviously, as you know, a gene that's mutated in some Parkinson's patients. That's the antibody that's been recommended by the Michael J. Fox Foundation. There is a signal there. On the knockout, you can see it. Oh, there she blows, right there. But clearly, there was nothing superb out there. So one of the companies learned about this. It was Thermo Fisher, and they said, okay, there's a market gap. Clearly, people will want it. And then they went and made that antibody. Actually, they made two, but that one's pretty darn good. Right? And it's renewable. It's a rabbit recombinant, right? So in this instance, it was as a community doing the experiment, identifying the hole in the market. Thermo Fisher went and used their internal resources to make an antibody. And now through this, they just say, here it is. And this is the best form of advertising, because this experiment was done by a third party, obviously, with no skin in the game, right? And that's, you can kind of see that the model doesn't work unless you do it open science. Unless you get 10 companies to competitors to agree to one reagent. You don't rank them. So, you don't, there's no profit motive for us to do it. So there's no advertisements, right? It's kind of a very unique situation where you solve a problem that's been befuddling the community for 35 years through this business model. And what we've learned so far is we've tested 389 against 34 proteins for those applications. 39% of the antibodies that the manufacturer on their website, on the catalog, say, hey, they're good for immunoblots. They don't recognize the right protein. 74 recommended for immunofluorescence don't actually recognize, the signal does not come from the protein. Of the 179 papers that used any of the antibodies, 99 did immunofluorescence with an antibody we know is nonspecific. This is incredible, right? And everybody who does use an antibody before knows this, but it's just quantifying it is something interesting. Some good news, though. No, they don't use immunoprecipitation as a metric, as a sort of standard test in the community. And we found 79 or whatever, 76 antibodies that really pull down the protein from homogenate, which is cool because then you can use these for protein interaction studies. So we're done, eh? What am I time? Oh, okay, cool. So now I'm going to talk about, the last one is, okay, that's cool. That's a service model. It tackles an important problem. And the problem was important, and open science is the vehicle to solve the problem, right? The same thing with the SGC. The problem was, how do we get to the long tail, right? There's no mechanism to do that. Why do we create a consortium that works in the open so we can get resources to work in the open? Remember, I guess what I'm trying to say, I'm not a fanaticist about open science. I said there are so many problems in biomedicine where it is the only way that I can think of allows us to tackle that problem. This is the one, drug discovery. And so, as we've talked about before, the main trouble in drug discovery is that we don't know enough human biology, period. It's not about intellectual property. It's not about industry. It's not about academia. We don't understand disease. And nowhere is that more true than in the neurosciences, right? I tell you, go to a neurologist and say, okay, Parkinson's, what caused it at your house? And they go, no, because I don't know, right? And we're trying our hardest, but that's the problem. And so if you think of it, the next step, so now you're taking, there's a new mechanism that you want to investigate. Let's say it's in Parkinson's. The first time you take a molecule into the clinic, into people, for that idea, inhibiting, PINK1 or something like that, you're really doing an experiment, right? It's research. It's not drug discovery. Because drug discovery, you figure it'll work. We just got to make the right drug. This is, we're testing the hypothesis if PINK1 has anything to do with Parkinson's. And if you frame it that way, as folks who are supposedly interested in creating knowledge for the public good, then you start to rethink, what is the use of that molecule, the tool that I happen to be taking into the clinic? Is it an asset that I want to monetize, or is it a tool to learn more about Parkinson's disease? And I would argue it's a tool to learn more about Parkinson's disease, because if we show that PINK1 is a relevant mechanism for Parkinson's, then people can start to invent the best drug that'll be the safest and the most effective, etc. But until then, it's just a crapshoot as to whether it's even the right idea. And so what's the role of the public in this, as supposedly, right, it is yours, knowledge generators. When we start to think about drug discovery, should we not think of that first test of the hypothesis in human as a research project, not a translational science project, right? Because translational science, how are you going to commercialize? That's not the aim. The aim in that early stage where we should be playing, which is knowledge generation, is find out more about human biology. Let the other folks who want to make drugs. That's their job, right? That's the job of someone who has a factory, right? And wants to run regulatory studies and do all the sort of systematic stuff you have to do at best on an industry, right? I didn't say we've done an industry because they have the knowledge or it should be done in industry. I said, if you're going to make a medicine and you have a factory and you have to go rigorously GMP, et cetera, it's best done in an environment that's probably industrial. Okay, but the research stuff, which we could do inside of academia, right, should be done by us, and it should be done in the open, because you only want one person testing that PINK1 idea. There's no point in 400 people testing it, or else you get a curve like this, right? The more we can spread our scarce resources across the planet on different targets, testing different hypotheses, the more chance one of us will happen to be right. And it is a wild ass guess, and no one's smarter, right? They're all guesses, but why don't we guess as a community, okay, and then find the right answer? But then it gets back to who's going to pay for it. And so we're trying to explore different models for how one could invent a medicine to test the hypothesis. And if it happens to work, it'll be a medicine, but at worst case scenario, it'll test the hypothesis. And the M4K Pharma, Medicines for Kids, I'm missing a K up there, it's trying to figure out a business strategy on how to do this. And so the same shtick as before, but instead of Icarus, M4K Pharma is 100% owned by Agora. The same model. We, as a community of scientists, are going to try and invent a safe, effective molecule that's got all the pharmacology to get into the brain, test the idea that this protein, it happens to be a kinase, I'll show you, is important for the disease, and find out what happens. So we're going to take another one of the asides and think about intellectual property and drug discovery. So on the top here is the trajectory of a typical patent. So you file your provisional patent application, and then you file your patent application. From there, you got your 20 years. Starts ticking, tick, 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 right? The patent gets issued here, but all this time here, you're taking the molecule and you're doing your preclinical studies and your scale up and your 2 animal talks, all the kind of stuff that you need to go to the regulators and say, this molecule is safe, can I please put it in a human? And you file your IND, Investigational New Drug Application, okay? And you got a whole whack of data that supports it. Then you run your clinical trials. Tick, 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 where is it? Tick, 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 tick. And you file your new drug application here. And your dossier that you submitted to the regulators now is even bigger because it's got how you manufactured, what are your safety studies, what are your controls on your manufacturing process, what are the clinical trial reports or the clinical reports from all, right? This is a ginormous document. The FDA or the Health Canada looks at it and approves it. You get this time back, right? At the end, that's your patent life extension. And on average, you have between 10 to 14 years of patent life after your drug is approved, where patent is an exclusionary, right? No one else can market or make money off that drug. Well, in 1983, Hatch and Waxman said, oh my God, they're American senators. No, they were congressmen. Said, drugs are too expensive. We got to get the generics to be able to compete better. Prior to that, if Pfizer invented a drug and the generics wanted to compete, the generics would have to run that entire process again to get to market and prove to the regulators that it was safe. And a lot of times they said, it's just not worth it, right? Because it'll take so long and so much money, there's no point. And so they said, how do we get the generics to get to market faster? I got an idea. You know that big thick dossier that Pfizer submitted with their drug? All the generic will make you do is to copy some of it to be equivalent to something in Pfizer's data. You don't need to do all the studies. You need to show your compound has the same characteristics. The compound is this, the compound is that, and then we'll approve your generic drug. Okay? So they have great, that's a faster way. They called it an ANDA, abbreviated NDA. And then I thought, cool. Then they thought, oh shit, we got a problem here. Because as soon they'll let Pfizer go, and as soon as their drug's approved, the generics are going to sue, try to overturn the patent, and even if they're successful 1 every 10 times, they can enter the market right away. Thank you, Pfizer, spending all that money. I can come to the market right away. So Hatch and Waxman said, oh damn, we got to somehow protect against that. So we'll go back to Pfizer and say, Pfizer, you know that dossier you have? It's confidential to you for a period of time so the generics can't peek at it. And that's called regulatory exclusivity of the data exclusivity. That data package that Pfizer submitted belongs to Pfizer in America for 5 years if it's a small molecule. It's longer in the EU, in Canada, and Japan. No one cares about Canada or small market, but I put it up there just out of interest. And so this regulatory exclusivity is actually what keeps generics away. Because generics don't sue until their regulatory exclusivity is finished. Because if they sued beforehand and overturned the patent, they can't see the data. So they just go waiting for their regulatory data to become publicly available, then they can enter the market. So they don't bother to sue until like day 1 after the regulatory exclusivity ends. So think about that. Pfizer did all that work getting a patent, but what's keeping competitors away isn't the patent. It's the fact that no one could see their regulatory data. And that's when the generics enter the market. So it's stronger actually than a patent. It's a whole bunch of other advantages. Now, often it's shorter, but often it's longer. And for neuroscience, like in Alzheimer's trial, where the trial takes such a long time, you've used up your patent life by the time you get to market. So actually in certain diseases like that, it's the regulatory exclusivity that the CFO of the company will monetize. Now, of course, I'm being a little bit simplistic. What a company will do is they'll take the patent on the composition of the compound and then make the patent on the yellow version, the slow release version, and they'll put a humongous pile of patents around it, all that are useless patents, but every one costs money to overturn. And so that's what happens, right? So if you look at Humira, which is a drug by AbbVie, they've got 200 patents. There's one important one, that's the Humira. But then they got all these other ones creating a patent sort of thicket to stop people from suing them. Okay, so there's a little bit of weirdness in the business world, odious weirdness, but it's still there. So it's not as simplistic as there's a patent and regular. But I think the point from this one is regulatory exclusivity, like patents, they shield the product from generic competition. They're consistent with open science. You don't need a patent. All you need to have it rights to your data. You can share them and still retain rights, right? And then that can be your asset if you want to protect your market. Now, if you're going to charge as low price as possible, as we hope to do, there's no reason to compete with the market. But nevertheless, you have a period of thing where you're the only person allowed to sell it. It's costless to get because it's an act of Congress or our Justice Department. Not like patents that can be overturned, right? You just get it. That Skrelly guy, remember the pharma bro? What he did was he took a drug that had been on the market but had never gone through the process, paid for it to go through the process, and got his orphan drug exclusivity, which means he was the only one allowed to sell it. And then he jacked up the price like insanely to odious and egregious levels. Obviously, he went to jail, but so the regulatory exclusivity is that strong, right? Because he had the right to do that, the legal right to do that. It's not subject to patent, to challenge. You'd have to overturn a law of Congress. And it provides a period of market protection that's certain ex ante. So my lawyer wrote that one because I didn't know what that meant, but now I do. So in other words, when you're starting the drug discovery project, if it's Alzheimer's, when am I going to go to market? I don't know. 12 years. How do you calculate what your ROI is going to be if you don't know how long your patent life is going to be left by the time you're done? But when you got your regulatory exclusivity, you say, okay, I know after I'm going to have at least 5 years in America in a small molecule, 12 years if it's a biologic. It's really that important that in the Canada latest NAFTA negotiations, the last two things negotiated were milk pricing and data exclusivity on biologics. The Americans tried their hardest to get an extra 2 years of data exclusivity for antibodies because the Johnson & Johnson, Amgen, Genentech, they're American companies. So America was negotiating on their behalf to try and get them to be allowed to do premium pricing in Canada for 2 years longer. Canada thankfully fought back. But you can see, of all the trade agreement stuff, that was the most important to industry. Wasn't extending patent life or anything like that. It was getting more regulatory exclusivity. The Orphan Drug Act, what it gave you was more exclusivity, even greater power exclusivity. I won't go into it. Just and it's only 7 years. But as soon as that law was passed, there was a massive influx of cash. The patent scenario didn't change. All the government did was to give you, say, hey, we have this new form of exclusivity, and that was enough to drive capital into that area. There's about 40 new chemical entities in the Orange Book, which is the book of drug approvals that don't have any patent life, though clearly it's the regulatory data that we're providing the market incentive. And I just told you about this last one. So the project we applied this kind of thinking to, which is a work in progress, is a disease called diffuse intrinsic pontine glioma, and that's the survival rate there. It's a brain tumor is diffuse. So by the time you catch it, it's already over the brain. It can't be surgically removed because it's in the brainstem where you can, well, you guys know better than I, right? So breathing and eating and stuff. So it was kind of a lost cause, but then they finally figured out how to biopsy it safely. And what they discovered was that there's a activating mutation in some of the subset of the tumors in the ALK2 protein kinase. So from a drug discovery point of view, kinases are drug targets, and inhibiting kinases is doable. So we set about, why don't we create an open science ALK2 drug discovery program where we start from a compound in the literature and start to make it safer, effective, brain penetrant, all the kind of stuff that one would do. We ran the project as if we were in a biotech or pharma, the same rigor, the same processes, except every first day, Wednesday, every month, we disclosed all our chemistry to the public domain, right? We had a YouTube channel and we said, this is what we're doing this week, so that no one could patent it, make sure it was still in the open. We had contributions. I don't know, I was so afraid of my time that I deleted some slides. So after all that chemistry crap, we got to these compounds, 5 of them. We were funded to do 1 by OICR. We had contributions from Charles River through their social, social good thing of corporate responsibility. We had universities. Boehringer Ingelheim came, said, hey, I know that protein. We'd like to give a seminar at your Wednesday monthly meeting. They're a big pharma. And they didn't tell us about their chemistry, but they said, you know what? We looked and looked and looked for someone to do a good animal studies. Do it at Penn, Eileen Shore. That's where we would do it if I were you. That would have taken us 6 months to figure out, to do due diligence on each of the possible people. And they'd already done that. And they just said, and they wouldn't tell a biotech, right? But they would absolutely, this open science thing, gifted to the world, peace and love and everything. Okay, right? There's a very clear mission statement. This is to make an affordable thing, and they're clearly sharing their data. And sure enough, then we went to Eileen, who had money from the NIH to do studies. We said, would you do it for free? Right? We'll give you a first-in-class compound. You can publish it. You can use it in your NIH grants. You can do anything you want with it. Just you allow us to use your data, and you may not patent. Just I'm in, right? So she got what she wanted, was a first opportunity to use a cool new inhibitor in her studies. She got to publish it. And for us, we just got the study we wanted done because we wanted to see, is it effective or not? And it is. So we saw, over the years, we've seen various universities and private sector partners offer us stuff at cost. Overall, you know, we got 5 compounds that made our target product profile. We were funded to do 1. So we made more compounds than we assayed. We did more science with the extra money. Now, the issues we've encountered, and this is just in the last 2 years, it's relatively easy in our funding system to get funding for the stuff we did, right? New protein, new inhibitor, testing a hypothesis in a mouse. The next step is, how do you make 50 grams of that compound? No funder wakes up in the morning thinking, I'd love to fund that. That is so exciting, because it's usually done at a contract research organization. It's buying like a tub of this compound, a tub of that compound, and mixing it and purifying it and GMP. And so that we see that as a gap in the system. The Americans have funding for that, but it has to go through an NIH, which takes such a long time. So for nimbleness, that we're having trouble finding that mechanism. I think we have to work on that. Like I say, fewer options in the public sector for project management. This really requires someone on top of it every day. And students shouldn't do it. The postdoc shouldn't do it. A professor is not really our expertise. Really, you want someone to cracking the whip, organizing next Friday meeting. Do you have your compounds made? Did you do your assays done? Especially if you're in a distributed open science thing where Philadelphia is doing one experiment and Cambridge is doing another. Someone has to really be on top of that. In an industry, you'd have this project manager. That's his or her job. It's hard to fund that in our system. So that's kind of interesting. And then COVID, right? We haven't been out schlepping and knocking on doors to fundraise. So that's what I'm saying. I'm saying there is, in theory, an intellectual property mechanism to protect your asset, to protect your marketing rights. In theory, we could do it with open science. In practice, we can. We've never taken a drug from this idea from the beginning all the way to the end. DNDi for sleeping sickness has taken a drug at the beginning and paid for it with public funds all the way to the end. And it just got approved. It's a wicked, it took a treatment that killed 10% of the people to a pill that is saving everybody. And they did a deal with Sanofi where the pricing is embedded into the deal, right? I don't know if they said they did have a hard time finding a commercial partner for that. Someone would manufacture and distribute, do the post-regulatory stuff. So it's not impossible, but every piece is possible. We've not yet put it all into a systematic multiple times. And that's what we're going to try and be doing over the next 5 years, is to create a system where the North Star is access to medicine should be a human right. Let's figure out a business model how to get there. Instead of what currently access, medicine is an asset. Let's figure out weird ass ways to make as many people get it as we can, knowing that a lot of people aren't going to get it. And like COVID, we've seen what happens when industry is in charge of seeing who gets medicines. Even governments, right? Even governments, you know, the Americans are America first. They're not, oh yeah, here's Canada, here's it. So I think it's super important for us in all aspects of this to try and figure out a different way where medicines are more equitable, and the technologies too. And it's written up. I did a lot of fast forwarding through all the legal mumbo jumbo. So here's it. We wrote it up where it's got all the case studies and all the stories about the drugs that have been approved in this way and all the legal cases, etc. Thank you very much.
So thank you very much, Al, for a fascinating talk. We're over time, but I do want to give the audience a chance to ask a couple of questions. If anyone in the room, you can press the button at your seat. Any takers? Is anyone on Zoom? Okay, we have.
Behind you.
I'm on Zoom.
Oh, Steve Pellich. Yeah. So, yeah, go ahead, Steve.
Yeah, no, it's a great presentation, Alan.
Have you heard of kinases before?
Yes, I have. I think you know that. Your observation that most of these commercial antibodies don't work, I guess, is pretty well known in the field. I know we've tested about 6,000 at Connexus, and clearly about 75% of them don't work. Now, in the examples that you gave, you were more focused, I suspect, on pan-specific antibodies. And I'm just wondering, what's your success with phosphocyte-specific antibodies? Because they're lower in stoichiometry, they're often less in abundance, or we don't know what the stimuli is necessarily to get that phosphorylation. Have you had any success on phosphorylation?
No, we've actually on purpose stayed away from those. Similarly, the antibodies that are for methyl groups in epigenetics, everybody is asking for us, do you have a trimethyl lysine, blah, blah, blah. Those are, as you know, a lot harder. And because we decided to focus on the business model first to get the companies together, to get the sort of process and the funding scheme, we thought we should not choose the hardest scientific problem to start off on, and we work in later to figure out how to do what you're doing, which is much harder. Than we decided to be simple. We have little brains.
And are you able to actually generate the reagents yourself, or do you find that you don't want to be in a position to compete with the people that are helping to fund your organization?
It's not compete with. So the simple answer is no, we do not want to. The public efforts that have tried to do both, which is characterize and market, you see conflicts of interest emerge all the time. And so we thought it's safer for us to be clean, be agnostic about the result, and therefore gain the trust and not put us into conflict with any of the companies. And we think that's a safer way to go. And that's why we don't even rank them. We just sort of put the data out there and let the scientists decide.
Right. Well, I really applaud your efforts, and I'll let other people ask questions.
Any other? So I'll take one question, and then we'll wrap up if there's no others. So one of the arguments I've heard for patents is, even if it's not to make money, it's just that if you don't patent it, then no one will ever try to develop it. And me not knowing too much about this, I'm just curious what your response to that is, if there's a simple answer.
It is the most common question. And that's the legend that's been passed on from grandfather to father to this. There's no evidence of that, especially at universities, because we never invent stuff that's close enough to market to be able to do that not putting into the public domain would inhibit things. Yeah. And so, like I say, we're so early at universities. Our discoveries are rarely turned into products. There's millions of dollars in investment between what we do and what that putting in the public domain. Certainly, we have very specific examples for our compounds. There's 80 clinical trials that are ongoing now of ideas that we put into we got the compound, we shared it, they did discoveries, they found a idea, then the companies go, thank you very much, I can make my own compound, and off they went, right? So if anything, we spurred competitive drug discovery. Now, we didn't make any money, so maybe that's something that's the subtext to what you say, like how do we make sure we get a piece of it? So anyway.
Tim?
Yeah, really entertaining talk, and I think this was a great mission. I got a couple questions. I guess the first one is if you could just give us I don't know, maybe a short definition of this kind of regulatory exclusivity and exactly what that gets the company. I guess so that's a kind of protection that they have to market something into the future based on.
Yes, so it's a period, it's exclusive rights to the data that you used to convince the regulators your drug is safe. That very same data is what generic companies would need to refer to put their generic drug on the market. So if those data are secret, generics can't use the ANDA mechanism to get to market. So they have to wait until the regulatory period is over. Then in 5 years, let's say in America, it becomes available. Then the generics can refer to it and take their drug to market. Prior to that, they're de facto excludes competition. So Pfizer would be the only person to sell that on the market because generics can't look at Pfizer's data. So it keeps people away. That's the bottom line.
So it's a form of, I guess, kind of data protection.
Yeah,
and, you've invested in this.
It is intellectual property.
Right.
And it's
right.
And then it can be used for evil purposes. So Brazil doesn't allow it. So why would we allow a company to come into our country and say, no one else can sell this except you? With a patent, you have margin rights, you have a whole bunch of things. With this, you don't. And so they say, no way.
So second question is, you showed this graph with or image with all the logos and the companies that were willing to throw down 5 million euros and then get access to these structures and things. Do they have, is there some like privileged access?
No.
Like it's not like a gold membership or something?
No, nobody has.
They're just doing it to push the field forward.
Exactly. And pharma, of course, have big R&D budgets that they could, it sounds like a lot of money, but a $4 billion R&D, but they are some of the largest deals that they do. But they learn by doing, right? They don't get privileged access. They don't see the structures before other people do, but they do engage in the cutting edge of science, which is partially like if they we moved into epigenetics in 2010. Every company was going, should we go into epigenetics? I don't know. It's so big. We don't understand the area. If we join the consortium, we do it as a group. Then I don't have to spend $60 million internally. I can spend $5 million at the Sgc. I'll learn whether we should be doing it. If we should be doing it, then I can invest more internally. So that's the kind of decisions they use.
Do they have a role in identifying potential leads for future studies?
Yeah,
like a seat on the table.
Yeah, we just talk about it. Like what decision? So we decided we had about 3 different choices in this current phase, and we chose E3 ligases as let's do that one. And all of them said, yeah, that's the direction to go. And for us, so long as it's in the unknown, I don't care which one we work on, right? It's just a matter of doing one then the other. So.
You press the button on the mic.
Thank you for the talk. You mostly talked about the pros of open science, which I think I agree also. But I was just wondering, because you're practicing it, do you see any cons of open science, like anything you think might not be good because of open science, and putting that in context of international like politics and dealing with other countries which might not respect your IP. And
Well, it's kind of the opposite, right? If they don't respect your IP, it doesn't matter if you patent or not. I think it's harder work to share your data faster, right? So it is a lot of work to organize your data in databases or to make sure the reagent has a higher quality before you share it. And that seems to be the biggest complaint of practitioners. Oh my God, is an extras thing I have to do, right? And that's fair. But on the other hand, if you don't do it, what's the point of doing your research, right? If you're not getting maximum use out of it, the public's paying for it. But I would say that that's the number one. We haven't seen, of all the years, like twice we've been scooped, right? And we just didn't know. And because we're working on an unknown, just somebody happened to be ahead of us. But for 98 out of 9 times, we get more phone calls. Hey, I see you're working on this protein. Can we collaborate? Which is far more common, right? But I have a rich fantasy life where it's all going to be good. So I'm an optimist. So it is mostly the pain in the you-know-what in sharing. I would think that would be fair, right?
Okay, well.
Sorry for going too long.
No, thank you very much, Alan. And so let's thank the speaker one more time. If trainees here want to sit down with Alan and pick his brain more, there's a chance to do that.
That used to be my favorite part of seminars at McGill.
Yeah.
Because they had donuts, though.
I think we might have something.
I was going to skip the work stuff altogether.
Oh, glad you did. That was definitely one of our purposes.
Oh, okay. Yeah, because I had my slides set before you run in.
That's great. That's super interesting. So that's like the clinical trial data that's covered by existing.
Yeah.
And I read your article.
And GMP and, your talk studies, all that stuff. Yeah. But purely from a financial point of view, right? From an institution, donors tend to be local, so it's not like you're competing with the same donor pool as McGill. And it resonates really well, right? So you're going to see, and similarly, patient foundations tend to be local. It's a really cool story to